Welcome back to the Cambridge Neuroscience Interdisciplinary Seminar Series. This series features current work from the neuroscientists across the schools and departments of our university, reflecting the pioneering work and diverse interests of our members here. Each seminar focuses on one of the new interdisciplinary themes of Cambridge Neuroscience, which we will be launching later this year. For more info on the talks covered in this seminar series and all things neuro-related in Cambridge, please follow us on Twitter at CamNeuro and follow the links below.
So today I am delighted to welcome Professor James Rowe to our Cambridge Neuroscience Interdisciplinary Seminar Series. Disease-wise, James is interested in frontotemporal dementia, progressive supranuclear palsy, Alzheimer's disease, and he is currently the director of the Centre for Frontotemporal Dementia here at Cambridge and leads the clinical program for Parkinson-plus. So this is an interdisciplinary seminar series, so I'm delighted that James uses many different techniques in his research, such as brain imaging, psychopharmacology, computational modeling. And so today he's going to talk to us about apathy and impulsivity in neurological disease. So focusing on the causes, effects, and treatments. So welcome, James, and thank you for taking the time to come and talk to us today.
No, it's a great pleasure. Thank you for inviting me. And I'm aware it's a mixed audience, and I'm going to try and give a little taste of our range of work with something for everybody. There's some cellular work, there's some computational modeling, fair bit of imaging, some clinical slides, and some basic cognitive neuroscience. So we'll try and hopefully give some points of contact, whatever branch of research or clinical practice you're in. And hopefully, there we are. I've no relevant disclosures for today's material. I'm going to be slightly didactic for the first few slides, just to highlight that apathy is not a unitary entity. It's a sort of behavioral construct about what we see in other people. It's not around what they feel, but it's what we observe them doing, particularly in goal-directed behaviors. And it may encompass a lack of action, but we need to distinguish it from the akinetic lack of movement. Not acting without a goal is not the same as not moving, so we need to distinguish that. People may have an emptiness or a lack of feeling, a lack of passion, where the word derives from, but this is not around depression, and we need to work hard to separate depression from apathy. It may be manifest as a failure to be spontaneous and initiate activities. It may be more in terms of cognitive activity rather than physical movements. So hold that in mind, a sort of a picture of apathy. It's rather difficult to get good films of apathy because by the very nature it's kind of not doing things. So it doesn't make good coverage in film, but hold that picture of apathy. I'm going to ask you to contrast it initially with one's view of impulsivity, which again is a complex construct about people taking high risks or not considering rewards and outcomes, ignoring those negative outcomes as well as positive ones, failing to reflect and gather enough information before making a decision, and we'll return later to the formal process of evidence or information-based decisions later in the presentation. People may be very impulsive in terms of not tolerating delays or failing to inhibit inappropriate responses, so failing to rein in actions. And all of these get brought together in impulsivity, and with slightly different biological bases to these different aspects. And I think in the lay sense, you often think of apathy perhaps as being too slow and doing too little. And that's certainly in much of the literature been strongly associated with a low dopaminergic state. Whereas impulsivity, you might think of as being too fast, doing too much, too quickly. And that's also, impulsivity has been linked to a hyper-dopaminergic state, too much dopamine. So the literatures on apathy and impulsivity about motivation and risk-taking have a sort of contrasted ends of the spectrum, which I'm going to, again, say from my view is incorrect. Apathy and impulsivity are more around the quality of the decisions that people are making. And one of the pieces of evidence from this, that apathy and impulsivity are not a sort of Jekyll and Hyde opposites, comes from the data in patients with the diseases like in those videos we've seen. Also comes in normal populations and Parkinson's disease. And it's this observation that apathy and impulsivity are positively correlated. So instead of being opposite ends of a dopaminergic spectrum, too much, too little, too fast, too slow, overacting toward or underacting, they're actually positively correlated. In frontotemporal dementia, we'll see more today, progressive supranuclear palsy, Parkinson's disease, older populations, it's a recurrent finding. So they're not just opposites. So we need to think about what then are the drivers, what are the mechanisms of apathy and impulsivity, particularly in disease states. So I'm not going to go through all these papers, but if there's sort of students online, you want to freeze the recording just to pick apart some of the references, this would be a head start. But briefly, I'm going to say there is clearly a dopaminergic story to apathy and motivation and impulsivity and risk-taking, but it's only part of the story. There's a lot to do with the brain structures and how diseases cause atrophy or degradation, degeneration of key brain structures. There is a serotonergic story, which I'm not going to focus on today, because I really want to take us forward to the noradrenergic aspects underlying both apathy and impulsivity, and particularly the way that may lead us towards new and effective treatments. So to take this further, we're going to need data. We want data from representative populations. We're going to look at people who have a wide range of illnesses which are manifestations of frontotemporal lobar degeneration. So this includes frontotemporal dementia, progressive supranuclear palsy, corticobasal syndrome, and related diseases. And we looked in the study called PIPPIN nearly 10 years ago, trying to identify every patient in this sort of northern East Anglia region, and met most of 80% of these 200 patients. And we repeated the study 4 years later and are now midway through a third phase, trying to identify and look in detail at the symptoms of patients who have these disorders. And there's a few surprises that turn up. One is to say, if we look actually at what their symptoms are, and we measured around 80 clinical variables, the presence and absence, some of those I focused on here, the much more clinical symptoms. If you read the names, you'll get a sense of the sort of cognitive and behavioral changes we were looking for. We then looked at using a method called multidimensional scaling. If you have one symptom, how likely it is to have another. So what are the natural clusterings, the way that symptoms go together? And the closeness on this 2D plot represents the probability of two symptoms co-occurring. And one thing I want to highlight really for this talk is if you look down here at the bottom of this red cluster, is how close apathy and impulsivity are, highlighting that if you're manifesting high risk and challenging and impulsive behaviors, you are also likely to have apathy, okay, as observed by carers and those around you. The color coding in here for clinicians or those who work with patients with frontotemporal dementia is to bring out clusters that are reflected in the diagnostic criteria for each disease. And if you look at this blue cluster, very PSP-like, red cluster looks very frontotemporal dementia-like, but this is post-hoc color coding by me to emphasize that syndromic patterns. But actually, these clusters are in a continuous space across the sort of syndromic or symptomatic phenotyping. You don't get clear separation of patients. And even within that, apathy and impulsivity are closely related. So again, to look in a bit more detail, we measured both clinical variables and a range of questionnaires of the patient and relatives and assessment scales and some biomarkers with imaging and neuropsychological tests. Get a very large number of variables, which we wanted to reduce down in a simpler way through the principal components analysis to say, what are the clusters of symptoms and signs that belong together? What are in other words, the patterns and how do they relate to the underlying illness? So you might call these components or behavioral modes. And one of the things we showed is that the presence of apathy and impulsivity under this group, I'm going to call them challenging behaviors, but the really apathy and impulsivity comes together to a variable degree across patients. So each dot on this chart represents an individual patient. It's the extent to which they're manifesting the presence of this PCA component of challenging behaviors. And we can see that the values in red are high in patients who have been clinically labeled as frontotemporal dementia behavioral variant. That's the defining features of that illness. But it's actually equally high in about half of those patients with PSP and corticobasal syndrome, and many of those with semantic dementia, in whom it's not a diagnostic criterion, but they still manifest as apathy and impulsivity. And the extent to which you do manifest apathy and impulsivity has a huge impact on mortality. So one standard deviation increase in the expression of this behavioral profile is a near doubling of mortality at death 2.5 years later. Okay, so Whether or not it's causal, we can discuss later, but it certainly seems to be an indicator. And if we look in another way and we say, well, the amount of this apathy and impulsivity you have, let's look at your time, not just to death, but to nursing home needs or death. And again, whether you're in a high, medium or low group tercile for expressing these behaviors, adds around 18 months or brings forward around 18 months of time in which you need nursing home care. So again, for health, economic and family and social reasons, enormously impactful. The challenging behaviors, apathy and impulsivity across multiple syndromes caused by frontotemporal degeneration occur together and they worsen prognosis. We've separated out the akinesia and depression effects. We can perhaps talk about that if you like, but fairly clear these are not manifestations of depression. But when do they occur? Well, what I've shown so far is people with clearly manifest disease, patients, if you like, in clinic. But one of the features of frontotemporal dementia is it's strongly inherited. Around 40% of people have a genetic etiology, and half of those we know the gene. And that opens up the possibility to study members of their family 5, 10, 20, 30 years before symptoms come on, who are pre-symptomatic. This is part of what's called the GENFI study, Genetic FTD Initiative. And if we look at people who are in good health, no diagnosis, believe themselves to be well, and we measure apathy and impulsivity through questionnaires this time, in those who do not carry the gene, there's a very slow increase over the years, over the annual follow-up. But in those who are carrying a gene, who believe themselves to be well, have no symptoms, actually there is a subtle but more rapidly progressive increase in apathy. Okay. And if we turn this around and we say, based on what we know happens in your family, when do we expect you to get unwell? We'll call that time 0. We expect the date of onset for you and your family. And we track back in time. We start to see those who are carrying the gene to have a more rapid increase. And that increase, that diversion from the other members of their family who don't carry the gene, that divergence in terms of how much apathy you have is beginning 10 or 20 years before the expected onset. So subtle apathy begins early. Then there's a question of chicken and egg. Perhaps apathy is bad for your cognition. You lack cognitive stimulation, you're withdrawn from social groups. Perhaps apathy helps cognitive decline. Or maybe it's the other way around. Perhaps it's that cognitive decline leads to apathy. Okay. So how can we try and tease that apart? Well, one of the ways to try and tease that apart is through a longitudinal study and to measure both apathy and cognition and to look at the relationship and the predictive value of one of these on the other. So does apathy predict cognition or vice versa? So we're looking in pre-symptomatic gene carriers. This is part of the GENFI study, so it's a longitudinal phenotyping study, about 300 mutation carriers. They've got 3 big mutations, but roughly equal proportions. And we're going to compare those to 300 people in the same families of a similar age who don't have the gene. And essentially what we find is using, this is the work of Mara Malpetti, who's actually on the line at the moment. What we essentially find is that apathy predicts your future progression of cognitive decline, but not vice versa. So the details of that are shown here in these latent growth curve models. It's a form of path analysis for looking at longitudinal data. And it essentially says if we measure cognitive decline, so we're using a digit symbol test here, for example, around speed of processing and with a bit of executive function required over progressive years, we can look at the speed with which you slowly deteriorate on this test. And we can say, how does that relate to how apathetic you were at the baseline? And the slope, which is the progression rate of your cognitive decline, is predicted by your baseline apathy. So just to put this another way in the plot, I can't give individual plots because it might inadvertently give away the genetic status of individuals who don't know their own genes. So I've had to, we've got pooled data plots, which is why it's got the rather sort of bland format. But essentially, in gene carriers, there's a very steep relationship between how apathetic you are at baseline when we first meet you, and the slope, the rate of progression of cognition over time, which is not there in non-carriers. Okay. We can also use these models to ask, how does the progression of brain volumes in any given brain region one might consider? We looked at across the brain, how does progression of atrophy relate to progression of apathy? So brain to behavior relationships. And what clearly came out is the intercept, which reflects really the baseline, your pre-morbid atrophy state, or cognitive volume, I should say, before atrophy, that predicts the rate at which your apathy progresses. And it wasn't anywhere in the brain, it certainly wasn't everywhere. It was in two key regions of the anterior cingulate and the prefrontal cortex. So subtle pre-symptomatic atrophy in these regions predicts your development of apathy, and it's the apathy that seems to predict or herald cognitive decline. So it's important, it matters, it has an influence on your outcome. It begins even before symptoms begin, and we're beginning to show it has some anatomical relationships as well as some predictive validity. I'm going to take a bit of a jump now to think about some of the underlying mechanisms, bearing in mind that this is a complex construct, it's a multivariate problem, so I'm not looking for a simple single solution, but I am looking for solutions or explanations that can lead to treatments. So I'm going to talk now about the noradrenergic system of the locus coeruleus, and I'll circle back to the importance of that anterior cingulate and the prefrontal cortex in a few slides' time. So all of our brain's noradrenaline essentially comes from a small region in the brainstem called the locus coeruleus. It's small, it's diffuse, it's about the size of two little half-inch sticks of spaghetti, okay? And it's only got around 50,000 neurons in an adult human. 1,500, I think, in a rat, but 50,000, that's nothing compared to 10 billion neurons that would comprise your whole brain. Only 50,000 are scattered in this little segment at the back of the brain. And in those diseases where we see apathy and impulsivity, such as on the left here, progressive supranuclear palsy, or on the right here, Parkinson's disease, if we look at post-mortem reduced cell counts, we see there's a severe catastrophic loss of the number of cells in this locus coeruleus. It's also actually one of the first regions to be affected in the pathology of Alzheimer's disease. So in three very different diseases, it's affected early and severely. So we began looking at this a few years back, particularly in partnership with Trevor Robbins and colleagues, looking at whether behaviors like apathy and impulsivity were related to the noradrenergic loss and could be recovered or treated, at least partially, with noradrenergic drugs, focusing on this drug called atomoxetine, which is a noradrenergic reuptake inhibitor. It's well tolerated, very safe for human use with a few caveats, and does appear to be able to recover impulsivity or restore behavioral inhibition, at least in some patients. And that will become critical, which patients recover. So just to illustrate that, we study patients in the MRI with Parkinson's disease and a double-blind placebo-controlled crossover study with this noradrenergic enhancing drug called atomoxetine. During the task, people get activation of this medial prefrontal area that's somewhere between pre-SMA and anterior cingulate here, and the inferior frontal gyrus. So we'll remember these two locations here to focus on. And if we think about their activation in this task, in patients initially on placebo, there's a modest amount of activation. When we give the drug atomoxetine, it markedly increases. So we get more activity in these prefrontal regions, particularly this IFG region. Not in everybody. How severe your disease is correlates with the impact of the drug. So the more severe the disease, the greater the drug effect on the degree of activation. Okay. So the frontal cortex activation, which is reduced in Parkinson's, that activity is restored by the drug, at least in some cases. So we're going to need a precision medicine approach to try and say, well, who might those people be? How can we predict who might respond? So we come back to what I might call baseline dependency, the state of your locus coeruleus may, and whether you do or don't have a deficit in your locus coeruleus may predict whether you respond to the drug favorably. So if we look at post-mortem, we take a slice across the brainstem. This is from a brain donor into the brain bank. This is half of the pons. At the back of the pons, in the floor of the fourth ventricle, you get a little dot, okay? So that dark dot is the locus coeruleus. You can see it with the naked eye. You can see it because it's 50,000 cells in adult life over the age of 50 are dark under light microscopy, and they're dark because they contain neuromelanin granules. This is an iron-rich pigmented proteinaceous granular substance that builds up in the catecholamine-producing cells, including locus coeruleus. So the presence of all that iron disturbs the local magnetic field, okay, the degree to which hydrogen molecules will share their magnetic spin, or the nuclear magnetic spin, and that means we can then start to see it with an MRI scanner. So although one can see it in the naked eye post-mortem, it's a little bit late by then. What we want to be able to do by following this chain of causation is to get a picture in a scanner where this little red dot here corresponds to the little dark dot up on the top side here. I don't know if you can see my pointer. Sorry, let me go to the laser pointer. So this little dark dot here to the naked eye is what we see in lifetime in living patients. Here, this is the axial section through the pons, that bright dot. If we look in a coronal picture, this is that little stick of spaghetti, centimeter and a half long, is this red line here. So now we can quantify the locus coeruleus in living patients. This is very, very much better at 17 and 3T. So an ordinary hospital scanner really struggles to have the signal to noise and contrast for this. So the new 7T that we have in Cambridge is ideally suited. And indeed, if we measure that contrast to noise, the signal strength in the locus coeruleus, we see in blue healthy adult controls, so we see this reduction in Parkinson's disease and a more severe reduction in PSP, repeating what we saw post-mortem with that cell loss. So now we can measure individual living patients' locus coeruleus. And this is some work, very exciting work that was published last week and has some very nice press coverage, highlighting that actually the quality, the integrity of your locus coeruleus here in the brainstem, this is an extracted model of the brainstem from the MRI, the locus coeruleus little template shown in green, we've expanded it up here. So this is the locus coeruleus as a sort of 3D nucleus, and the loss of locus coeruleus integrity correlates with apathy. AS, I'm sorry, is the apathy scale. It's a questionnaire around apathy. So more apathy goes with a lower signal in the locus coeruleus. It's a negative slope, and that's averaged across the whole locus coeruleus. If we try and do it voxel-wise, looking at subdivisions, it's more in this caudal part of the locus coeruleus. So as you degenerate and lose noradrenergic cells in the tail end of your locus coeruleus, the MR can no longer detect it because you don't have that iron-rich pigment, and that goes with you being apathetic and having actually weaker cognition. Let's loop back to impulsivity, and I touched on that, the use of atomoxetine, a noradrenergic reuptake inhibitor, to promote noradrenaline. How does your response to noradrenaline relate to your locus coeruleus integrity? Well, the answer is it is related. That there's this correlation between the locus coeruleus integrity as measured on the 7 Tesla MR and your personal behavioral response to the drug in terms of impulsivity. We're measuring impulsivity using a stop signal reaction time task, and essentially having a shorter stop signal time is an improvement. You become better able to restrict or inhibit your responses, and those with a degenerate locus coeruleus have a benefit of the drug. Perhaps you'd picture that as the drug is restoring a deficient noradrenergic system. Whereas if you've got an intact locus coeruleus, then enhancing it, the noradrenergic supply further, is of little benefit and might even count against you. Okay, so those of you who work with dopaminergic systems in Parkinson's disease will be familiar with this idea of an inverted U response. There's an optimum, and either too little or too much is harmful. So if the disease gives you too little, but then the drug will improve you. If the disease has not yet taken a strong effect, then the drug may actually do you some detrimental effects on your behavior. So we're starting to be able to predict who might individually respond. That was on impulsivity. I'm going to just pause for a moment and say now we keep this link with apathy. So I'm jumping a little bit between apathy and impulsivity, although do remember that these two things are very strongly correlated. Apathy is a tricky thing to study. There are many questionnaires of apathy, and that earlier slide, 2 or 3 slides back, when we showed the locus coeruleus loss correlating with apathy, was based around questionnaires. Okay. But questionnaires don't really tap into mechanisms of apathy. We need to think, why is it that people with these diseases are apathetic? And it's not depressed. If you're asked about the mood, they'll say they feel fine. And I think it's worth perhaps thinking about, rather than apathy, there's an alternative Greek word of ataraxia, which some of you may have heard of. So ataraxia rather than apathy, I think is a much better term for what we're seeing clinically. And ataraxia refers to almost a blissful contentment with the world, that you are not perturbed by events and things around you. You're able to be detached from it, and it was seen in in ancient Greece as a positive state of detachment, even above apathy. But it reflected the idea you don't act, you don't respond to the world, you don't engage with it, which you might observe to be a lack of goal-directed behaviors. But it reflects content, a sort of form of a deep contentment. And one of the reasons you may be content is that you fail to recognize that the world is not as you want it. So I'm starting to introduce the idea here of a divergence between the world as it is, as you perceive it, and the world as you model it or would wish it to be. And that brings me to this task. We sometimes call it the Angry Bird task, developed by Frank Hetzmans, who's now at the Dondas Institute, where it's an effortful reward-based vision motor task, where we ask people to essentially to be able to launch a projectile. It's a bit like the Angry Birds task, you know, when you pull back on the rubber band and let the Angry Bird go and it flings across the screen to knock over boxes. Those who know Angry Birds. This is a sort of motor equivalent. You press a button, you have to generate a sustained force, and that releases or catapults the cursor across the screen, aiming for a target. And the dynamics, the physical dynamics of the task are very predictable and easy to be learned. And what we're interested in is not just how well people do it, that they get the Angry Bird perhaps close to the target, but then how well do they perceive their own performance? And people are biased. People typically interpret their performance as being more accurate than it truly was. It's like an illusion of superiority, and that corresponds to the prior weighting, the precision with which you represent the outcome of your actions. So if you have a very strong prediction, a precise prediction about what I think I have done, then even when the sensory evidence tells you you've made a performance error, you will be true to understanding your own poor performance. But if you have a weak precision, then your perception of your own performance is going to be more and more biased towards your goal. So there's a relationship not to the reward value, but to the precision with which you represent the outcome of your actions. A slightly different, related but different concept. And we've shown that through psychophysical modeling of this task, where you can estimate the precision of the prior, it correlates with apathy. And this holds true for young, healthy adults who need slightly larger samples in a population, but also holds true for patients with Parkinson's disease. So this starts to put more of a mechanism, a cognitive mechanism that will relate to the neurobiology in a moment, a mechanism by which you fail to act in response to the world not being as you want it. So I might again come back to this lovely word of ataraxia rather than apathy. The point about this is that not only does this precision of the representation of your outcomes vary with apathy, it also varies with the integrity of the locus coeruleus. And for my purposes, it predicts how well you respond to atomoxetine. So atomoxetine, this noradrenergic drug, changes your behavior. It changes your behavior by altering the precision with which you're representing your action outcomes. Okay. And we can see this by the psychophysical estimation of this prior weight, the precision of the prior against the effect of atomoxetine in relation to this measured 7 Tesla measurement of your locus coeruleus integrity. So starting to pick apart, why do some people respond to atomoxetine in a different way? And indeed, this relationship between the drug effect and your locus coeruleus integrity as a predictor of drug response, it's a rather complex high-order interaction, but such as it is, this relationship was most significant in people who were themselves apathetic. So this emphasizes the importance of the state of your baseline locus coeruleus and behavior to predict the drug response. And you might think about using that to say, who might I want to treat? Someone with apathy, yes, and with a degenerate locus coeruleus. And that's an individual whose behavior is likely to be improved. So the noradrenergic treatment, it restores this mechanism of goal-directed behavior, which is not the magnitude of the reward, which I would accept is principally dopaminergic related, but the precision with which it's represented, which I would argue is under noradrenergic regulation. So I talked about locus coeruleus, but again, it doesn't say how is this locus coeruleus doing it. And I want to return to those cortical centers we saw earlier, this medial frontal cortex and the inferior frontal gyrus laterally. The effect, we showed earlier in that Parkinson study, that the effect of this drug atomoxetine is related to their connectivity of these regions to each other and their connectivity to the subthalamic nucleus. And that depended on the state of your disease. So again, what kind of shape you're in, your condition of your brain will determine the effect of this drug on your behavior. And the connectivity turns out to be important. We're going to then look at a slightly larger study, not of drug this time, but the natural integrity of the locus coeruleus. This is work of Alessandro Tomasini and CamCam, where we're going to look at two regions of cortex, so the pre-SMA, so really in and around this region that's active in the task of response inhibition, and this inferior frontal gyrus on the lateral surface, also active in the task and deficient in patients. So, and these two regions are correlated or coupled, they're connected, you might say, and we can measure that connectivity with fMRI. So we can then ask the question, although we know that the locus coeruleus integrity correlates with behavior, what's the link? It's not going to be a direct link. We propose that the locus coeruleus moderates the connectivity between prefrontal cortical regions, the IFG and the SMA. We've shown that connectivity is restored in the Parkinson's patients with atomoxetine. In healthy controls, perhaps just your resting connectivity is a mediator between the locus coeruleus changing connectivity and that connectivity being related to the behavior. And that turned out to be the case. So in a mediation or moderation analysis in patients at CamCam, we showed that this indirect pathway between the locus coeruleus and behavior was best explained by it being mediated through the changing connectivity of the prefrontal cortical regions. We can then take that to a lesion and say what happens if you have a lesion in a patient here with this medial prefrontal cortex. We can show behaviorally that this patient in red had a much longer stop signal reaction time. They were impulsive, very different from a control population shown in blue. This is an area, this is a lesion that takes out one of these critical cortical regions, and that region that was part of this mediating network. How this takes effect, we come back to psychophysicals, this computational modeling, and we can say that a decision to go or to not go is really a decision between two boundaries, two potential outcomes. And you may start either with equipoise or somewhere in the middle, and over time, your neuronal populations accumulate this decision variable from a baseline starting value up to some threshold. It might be an upper threshold to go left or lower threshold to go right, maybe an upper threshold to go or lower threshold to not go. But essentially, this gives us a way of evidence-based accumulation for decisions about whether I act and what action do I choose. And we can psychophysically sort of decompose the behavioral profiles to estimate the parameters of this formal decision. And indeed, this patient with the focal lesion, the way that interfered with behavior and made the patient impulsive was by reducing the threshold of evidence required for them to commit to a decision. So the decision boundaries came closer together. So the decision becomes more vulnerable to stochasticity, to random chance. But interestingly, the rate of evidence accumulation didn't change, but the threshold at which you acted did change. That struck us very interesting because when we come back to those impulsive conditions of Parkinson's and PSP, where on a go, no-go task, they are similarly impulsive, so they make a similarly high number of commission errors compared to older controls. This simple behavioral measure of accuracy doesn't tell the two groups apart. We decompose their behavioral profiles into these formal decision models about whether one should go to an upper boundary or to not go to inhibit behaviors. And when we look at this in patients with PSP, that impulsive condition with impulsivity as well as apathy or Parkinson's disease, what we find is that there is a change in that relationship to the decision boundary. And specifically, the effect we found was in PSP, I'm going to summarize this in red, whereas control subjects in black start out their decision in near equipoise between going and not going, and then can accumulate the evidence, if you like, the guiding evidence rapidly as to what should I do. They accumulate that evidence rapidly to go. Patients with PSP cannot accumulate that evidence. They have a deficit of getting towards a boundary, but they compensate for that by starting with a very strong go bias. So two things will happen, or several things will happen as a result of this. One is that although they're profoundly akinetic, they're paradoxically biased towards going, to a motor recklessness and impulsivity. Okay? So that explains how you can be both akinetic, but you're starting close to the boundary. What they also have difficulty with, though, is gathering further evidence to make a decision. Okay? So it takes a lot more to drive that decision evidence towards a boundary. And this may partly contribute to apathy, failure to accumulate evidence. But it also means the quality of the decisions are poor. If you have a shallow slope but a near boundary, you're much closer to a sort of roll of the dice and sort of noise in the system. So the quality of the decisions are going to be impaired. Whereas a control participant, healthy adult, starts out in equipoise, can gather evidence such as it is quickly, but has to have good quality evidence to either go or no go. So we're starting to see an integrated framework about how it's the quality of decision to go or to not go that links both apathetic and impulsive behaviors. And I would say this is under neurogenergic control mediated by connectivity. So what about neurogenergic strategies? Well, this takes me back, and I have to say I must thank Barry Everett, who's online for my whole career on this. It was Barry that got me into neuroscience 20 something years ago for my undergraduate part 1 here in Cambridge, and then into my part 2 I worked with Trevor Robbins, and this is my undergraduate project from many moons ago. Interestingly, also on this drug idazoxan, a neurogenergic drug, looking at attention and decision making in rats, to be fair, at that time. And we fast forward with the inspiration from Barry that I've never forgotten, and Trevor, who I worked with more closely in subsequent years, to come back to trying to target this adrenergic impulsivity pathway, both in Parkinson's disease and also increasingly in Alzheimer's disease, where, as I mentioned, you get a very early loss of the neurogenergic supply. So this is not my trial. This is a trial of a neurogenergic drug showing in people with early Alzheimer's disease an improvement in apathy. And this is not published yet, but I'm very excited by this work in collaboration with colleagues at Imperial and UCL and Oxford, and which will be out shortly, looking at a meta-analysis of various neurogenergic drugs, a wide variety of neurogenergic mechanisms, but in meta-analysis, they combined to have a significant effect on apathy. So this is from clinical trial data. We wanted to bring this back to these very impulsive conditions like progressive supranuclear palsy and frontotemporal dementia, and are now partway through this clinical trial called NORAP, so noradrenaline for progressive supranuclear palsy. So we've come all the way from those rats many years ago through some observational psychopharmacology, enriching that with imaging-based tools to understand how and why individuals vary in response, and now in a phase 2b clinical trial. This is a symptomatic treatment. But of course, remember that where we began, that apathy and impulsivity predict nursing home timing and your death. Yeah. So although this trial is based around symptoms of apathy and impulsivity, if apathy and impulsivity are causally linked to your risk of mortality and independence, your nursing home and death, then actually a symptomatic treatment could also be a disease-modifying treatment and improve your survival, your prognosis. So that's very exciting for me, and I'll be able to take further questions, but I'd just like to wrap up with some summary slides of where we've been. Well, I showed you that apathy and impulsivity, despite their complexity, are also positively correlated. They worsen prognosis in multiple diseases. Okay. They begin pre-symptomatically, and the apathy seems to predict cognitive decline rather than the other way around. I jumped to make a link to the role of the locus coeruleus and the catastrophic impairment of the locus coeruleus in Parkinson's disease, progressive supranuclear palsy, also Alzheimer's disease, but I didn't show you the data for that. And I think the locus coeruleus acts to support these behaviors through modulating the strength of connectivity in prefrontal cortical regions that mediate or make decisions. And you can think about this in formal decision models, either around the precision of the outcomes that we expect from our actions or their impact on decision boundaries. So we try and get some psychophysical modeling to understand individual behaviors. But for all of that, cognitive neuroscience and psychophysics, essentially we want to move this towards treatments for apathy and impulsivity, and with a strong expectation that whether you respond will depend on your locus coeruleus integrity. And indeed, this NORAP's trial is building in an imaging arm to see if we can get some precision medicine predictions on who will respond. So with that, I'd like to thank you for listening. I'd like to thank the superb team. This is our pre-COVID team meeting. I look forward to us all gathering together imminently. Many people who have actually done the work underlying this, and to you for listening. So thank you.
Thank you, James, for that wonderful talk. Join us next time for the last episode in the current series when we welcome Professor Sir Simon Baron-Cohen, a professor in the Departments of Psychology and Psychiatry and Fellow at Trinity College. Simon is the Director of the Autism Research Centre here in Cambridge. He has published over 600 peer-reviewed scientific articles, written many books for parents and teachers, and has made tremendous contributions to many aspects of autism research. He will be speaking on the prenatal sex steroid theory of autism. Don't miss it. For more info on the talks covered in this seminar series and all things neuro-related here in Cambridge, please follow us on Twitter @CamNeuro and follow the links below. See you next time.